So one way we can do that is to measure these small electrical currents.
You can actually measure the current in a neuron.
You can measure the current in a neuron, and all you do is, if you have an electrode, so this is just a little glass pipette that you can put into the cell.
Into the neuron.
Into the neuron. No. It has a little electrical wire. So it is in contact with the cell, and any electrical current that the cell passes will be caught by this electrical wire. And that is just hooked up to an amplifier, a normal amplifier. An amplifier is a device that just magnifies the size of the current.
Okay, so you can see the currents.
You can actually see the activity of the cell. So that's what we do. We can look at normal cell function. We can put a drug on, and we can see how the drug changes the activity of the cell. So we can do that in brain cells. Okay. So we can brain in native brain cells. We can do that in what we call cultured cells. So these are cells that are just grown in a lab. They're not taken from a mammalian brain. And we just keep them alive in a normal incubator, and we can record as well. And then there's some other cells, again, that are cultured, and they are unique because they don't express any proteins.
Oh, right.
Okay. So, for example, oocytes, so the frog eggs.
Frog eggs. Yes. Frog eggs don't have proteins.
They don't have neurotransmitter proteins.
They don't have receptors on their surface.
And they're not electrically excitable because they do not have
because they don't have those ion channels.
Exactly.
Because they're just basically dividing and turning into a frog.
Exactly. But they are unique because they're rather huge. And if you inject a protein into there, like a neurotransmitter receptor, it will start making that protein.
Right. So it has all the machinery to make the receptors.
Exactly.
It just doesn't normally do it. But what do you put into them to make them do it?
So we put in the instruction to make a protein, which is the messenger RNA.
Oh, okay.
Okay. So we put in basically the gene product.
Right.
Instead of putting the whole gene, we just put in the gene product, which is the messenger RNA, and that cell will happily start making proteins.
So you can take this cell, and you can put into it a protein that makes a GABA receptor.
Exactly.
And you can actually put in a specific RNA to make a specific subtype because there are many subtypes of GABA receptors.
Yes. Yeah, absolutely. And importantly, we can target the human form of GABA receptors.
Oh, so you put in the human mRNA?
Yeah, the human construct. So we know that.
Do you take that from humans, or is it made in the lab?
No, it's all made. It's just made in the lab.
So people know the gene, and the gene is different in the human, say, than the cow or the frog. So you get the human gene, and you make the mRNA.
Yeah, exactly.
You put it into the egg, and that egg then grows a lot of these special receptors that we're interested in.
Absolutely.
And then you can look to see whether GABA-A or Sentia or specific ligand, specific compounds work on that particular receptor subtype.
Absolutely. And that is what another researcher in my lab has done. So
how do you actually do that? I mean, how do you actually measure the activity across these?
So this is work done by another researcher in my lab, so Rita Montero. And so she's made the mRNA. She's injected it into these cells, the oocytes. And after a few days, we know that they start making the protein. And then she used a technique called 2-electrode voltage clamp.
Okay, that sounds complicated.
Okay. So it's exactly as it sounds. So you have two, instead of just having one electrode, where I was measuring the currents. Now we're measuring the voltage, which is the potential difference between a cell.
Right. Oh, inside and outside.
Inside and outside the cell. And why is that important? Because a cell is polarized. A neuron is polarized. Inside is negative, the outside is positive. So if you change the membrane potential of a cell, you will induce certain currents.
Right.
Okay. And that is what's happening right now in your neurons.
Right.
It's firing action potentials and certain currents are moving in and out.
Okay.
What we do is we have two electrodes. One is measuring the voltage and we clamp it, meaning using an amplifier, tell the cell we're going to keep your voltage at a certain potential difference.
Right. Yeah.
Okay? Let's say minus 60 millivolts. We then have another electrode that is measuring the current that might be passing at that time when we're clamping it at minus 60. If you then apply a compound that opens certain ion channels, your cell is going to want to change its membrane potential. But you clamping it.
You make sure it doesn't.
You make sure it doesn't. Therefore, to do that, you're going to get a change in your current. You get a measure of your current. Yeah, okay. Exactly. So that's what we're doing. We basically clamp it at 60. And if you apply GABA to your GABA receptor, we will see this current that comes in.
Because, let's go through this slowly. So you've got your cell and you've got your special, each one, you can choose your receptor subtype.
Yes.
The cell grows it. Yeah. And then you got your electrodes in and then you drop GABA on and then the receptors open and ions go into the cell and you measure the number of ions, effectively, the magnitude of the ions.
Exactly. So basically the electrode quotient of this ion movement, because we are keeping it at that holding voltage. And you can, the beauty is that it's quantitative. So if you throw more GABA on, you'll see a bigger current. Okay.
So GABA does that, but so do these things.
Exactly. So that proves, because if we did not put in the mRNA, if we just put GABA on an oocyte, you would get no current.
Right.
So that tells us that your current is due to GABA, and it tells us that the cell is making your receptors. Right. And there's no other receptors in there.
Because you've just put in the one RNA for that one receptor.
So now, if you put on anything else and you get a current, not GABA, anything else, you know that compound is working like GABA. It has GABA-like function.
And you've done that with Arcarel.
That is what we've done. So we've done it with Cynthia, we've done it with GABA.
And with our synthetic molecules as well.
With your synthetic molecules. And we see these beautiful GABA currents, proving unequivocally.
I can say they are beautiful, actually, because the fact that they're dose-related is really elegant, isn't it?
And the important thing is that when you then pharmacologically block the receptor with your antagonist, so it's blocking it, you can actually prevent the activity. So that is the industry standard of proving that something is GABA, acting like GABA. Unequivocal proof.
And because you have put in the human receptor, then that's that little egg is a proxy for the human brain.
Exactly.
That's very impressive.
Yeah.